thank you, first of all, for attending this evening and to welcome everybody who's come along to this second Gifford lecture in the series of 6 that are being presented this week and next. So as was explained yesterday evening, for those of you who were at the first lecture, the Gifford lectures were established at the close of the 19th century by Mr. Gifford, and they were bequeathed a sum of money to run these lectures at the 4 ancient universities of Scotland that dealt with natural theology. And of course, these days, natural theology, I mean, with the growth of science and philosophical understanding as well as working theology, natural theology is a tremendously rich field, and the university has a group that thinks about who should be invited to be the lecturer. We've had some tremendous lecturers in previous years. Sarah gave a list yesterday evening, but we've had Iris Murdoch, Mary Beard, Stephen Pinker, very many others. And this year also we have an excellent lecturer in John Dupré. Also, we have a Gifford blog. So there's a blog that's already been started by Victoria Turner, who's the social media host for this. And if you would like to join in the conversation about the topics that have been raised by John's analysis of the process view of life and this evening of evolution, then please feel free to participate in that blog and take the argument further and ask your own questions, state your own opinions, and participate and keep the conversation bubbling along, even on the days when there isn't a lecture. Now, the final thing that I want to say is a little bit about John Dupré. So yesterday, the introduction covered John's many academic honors, the visiting positions he's held, and so on. I just want to say that I know John because in the noughties, towards the end of 2006 onwards, John and I were directors of sort of companion ESRC research centers. There was a network working on social science and philosophical aspects of genomics and of the genomic revolution. And so John was the head of the eGenis Center at the University of Exeter. And I really just treasured those times with John and the intellectual activity that he cultivated there. In some ways, the center that he led was the one that was the most unexpected and in many ways did many of the most novel and exciting things. And I've always taken that as a very direct reflection of John's systematic and inquiring approach to these topics. I also remember John stepping in last minute to help out with a talk at one of the conferences we organized, and with very little notice, producing a simply brilliant conversation around synthetic biology and the social management of the new biological opportunities. Now, this evening, John is going to take his process view of life further to introduce what he thinks this does for changing our understanding of how we think about evolution. So yesterday evening, John introduced the, as it were, getting away from thinking about things to thinking about processes, introducing this process view of life, and said essentially that everything may be a process, but certainly within biology, things are processes. And so this evening, John is going to help us understand how evolution is best understood as a process. So if we could offer a welcome to John as he gives us this talk. Thank you very much, John.
Thank you very much for this very nice introduction. Okay, so in the last lecture, I described a contrast between a philosophical orthodoxy that sees the world as composed of things and an alternative view that sees everything as process. What we tend to think of as things are, I claim, no more than temporarily stabilized structures in a world of change or process. I illustrated the point with a discussion of the nature of the organism. Today I turn to the most celebrated scientific idea in biology, evolution. No one doubts, of course, that evolution is a process. Evolution is a theory of change, of how the immense diversity of our current biosphere came into being from vastly simpler and more homogeneous origins. Nonetheless, a processual perspective can have profound implications for our understanding of evolution. Let me begin by getting clear about what it is that evolves. We sometimes say casually that humans evolved, but of course no individual human evolved. Humans develop, mature, and eventually die, but they do not evolve. What evolved is the lineage which humans now constitute. But there are some subtleties to the concept of a lineage that I want now to explore. It will be good to start with the very familiar image of the tree of life. Let us assume for the time being that all species are sexual and that there is no hybridization. The tree of life represents the history of life. It is widely agreed that all living things, with the possible exception of viruses, which many biologists consider not to be living anyhow, derived from a common ancestor, sometimes known as Luca, the last universal common ancestor. The tree of life can be drawn at many different scales. In this slide, we see images with increasingly fine detail of human evolution, either of which would be a very small detail of the element labeled animals on the previous slide. The first shows the evolutionary history of primates, the second of hominins, that is, all extant and extinct human species, with chimpanzees for reference. A complete tree of life, certainly not something we will ever be able to draw, would have all the details of this third picture, which almost certainly excludes many as yet unknown extinct species, on every part of the map. The lower parts of the first large-scale map would be covered with great subtrees of now extinct species. But note that however much we simplify the tree by increasing its scale, any line on it represents what was once a species or a sequence of species. If we look at lines near the bottom of the large-scale picture, the length of line that remains the same species will be very short. All the branches that are not shown represent points where the species might end. For cladists, who are now the dominant school in biological taxonomy, a branch is by definition a point at which one species ends and two others begin. With the tree of life in some focus, we can now introduce two key concepts that are sometimes confused, the lineage and the clade. The clade, which has become an entity of central interest to taxonomists, the classifiers of living diversity, is a species and all its descendants. At whatever scale we draw the tree of life, if we cut through any of its branches, what we thereby detach is a clade. So this line detaches the hominin clade. The clade, then, is forward-looking from a species to all its descendants. Cladists believe that every taxonomic unit should be a complete clade. Lineages, however, are backward-looking. A lineage can be found by starting at a species and tracing back through the sequence of species that preceded it, in principle as far as Luca, the universal ancestor. I noted that lineages and clades are sometimes confused. If you search on Google for images of a biological lineage, you will find mainly pictures of clades. Definitions of the lineage are often forward-looking, though sometimes including a word such as line that points to a more accurate understanding. But the backward-looking definition is far more straightforward. Going backwards from the outer reaches of the tree, there is never any question which way to turn. You should always go backwards in time. Of particular note in thinking of a lineage this way is that at every point the lineage is precisely coextensive with a species. This is also why it is useful to restrict the account in the first place to sexual species. A sexual species, by virtue of actual or possible reproductive links between its members, can be seen as a coherent and persistent process. Hence, the lineage, as an ancestral descendant sequence of species, can be seen as a continuous process. evolutionary change, finally, can be seen in the differences between the properties of the species at one stage of the lineage and the properties of the species at a later stage. There need not, of course, when I say the properties of the species, I of course mean the properties of the members of the species. There need not, of course, be any sharp transitions between species. In the cladistic taxonomy, I'm currently assuming a new species appears simply because the lineage encounters a new branch in the tree of life. That's to say, a division in the evolution. It may be that the origination of a branch requires some kind of significant change, but that may be located in the other branch. At this point, one might relax the assumption of no hybridity and even, relatedly, allow lateral gene flow by other means, such as viral vectors. Lineages are often thought of as vehicles for gene flow, but introgression of genes from other sources is not necessarily a problem. Indeed, it may just be seen as a significant potential origin of evolutionary change. Hybridity presents a slightly different problem. Suppose that two closely related species have a lot of gene flow between them through the production of fertile hybrids. Why should we think there are two species rather than one? The answer is that there is no reason. Fortunately, this is not a problem for a process ontology. Processes do not require the sharp boundaries assumed for things, and counting of processes may often be a matter of arbitrary stipulation. In the present case, history may eventually decide the matter as the lineages do or do not diverge. Retrospectively, there may be no question whether they have become distinct, but there may be no objective answer to the question when they became distinct. The promiscuous lateral gene transfer found among bacteria, or at least some bacteria, presents a more serious problem. Here is an image of the tree, or better, net of life by the evolutionary microbiologist Ford Doolittle. Note that there is no unambiguous way of tracing the lineage back from a point on this tree, as there are mergers as well as splits in the forward evolution. So when you're going backwards, you come to a point where there is not just one way of going backwards in time. Putting the same point in a different way, lateral gene transfer means that even knowing the direct cell line ancestry of a cell will not provide complete information about its genetic ancestry. Moreover, since microorganisms are asexual, it is anyhow much less clear what constitutes a lineage. Any cell seems to have an equal claim a priori to be the founder of a new clade, and many cells will be parts of large numbers of lineages. As a result of these problems, there is considerable controversy whether there are coherent lineages among bacteria and other asexual single-celled organisms. Once again, this is not a problem for process ontology. We have a mass of cells dividing and mutating and transferring genetic material. There is strongly stabilized structure at the level of the cell and the single-cell lineage. At the level of the species lineage, there may be none. Some scientists have recently suggested that most bacteria do belong to species-like groups, exchanging genes very largely among themselves. If so, there are indeed real species-like lineages. This is an interesting scientific issue, but not one that really touches my present concerns, though I will return to some of these issues. I've already addressed the first question that arises in giving a proper account of evolution: what evolves? The answer is lineages, species, or sequences of species. Evolution occurs when distribution of traits in a lineage at one time differs from the distribution at a later time. Are species evolving lineages? With one minor qualification: yes. Many species consist of a number of more or less isolated population. The rabbits in Australia, which may still count as belonging to the same species as their European ancestors, may not be part of the same reproductively linked community, though they do no doubt still maintain at least possible reproductive links. And I'm sure the odd pet rabbit gets taken to Australia and escapes and so on. We may treat this problem much as we treated hybridizing species. If the species stay apart indefinitely, they will surely diverge to the point that they become different species. If they reunite, then they either will or will not re-merge. So retrospectively, we can decide the question. At the present time, we may say what we like. The listener will have noticed that I've been speaking of species as a kind of entity. In fact, in my view anyway, a continuent process. But it's also common to think of a species as a classificatory or kind term. Rabbit, one might think, was a general term of which Peter Rabbit, Roger Rabbit, and Flopsy Bunny are singular instances. So can it also name a singular entity, the process that is the rabbit lineage? In fact, as I shall explain a bit later, I think it can. But a philosophically less puzzling proposal is to suggest that what Peter, Roger, and Flopsy have in common is not being members of a kind, but rather being parts of a lineage. This also accords, I should perhaps say these are not the Peter, Roger, and Flopsy that you know best. These are real live rabbits by the name of those names. This also accords with the fact that given the points noted in my previous lecture about evolution and variability, it may be difficult to say what the criterion could be for membership of the rabbit kind. I've argued at least that there is no essential property of rabbits that could serve this function. For now anyway, let me say more about lineages, including species, as individuals. Philosophers of biology will know that the claim that species are individuals was a notorious thesis proposed by the biologist Michael Gieselin and the philosopher David Hull in the 1970s, and eventually quite widely accepted. One reason I say notorious is because philosophers, at least, had taken terms like rabbit, horse, or oak tree as paradigmatic kind terms for over 2 millennia. To be told that they were proper names of individuals seemed rather shocking. On the other hand, some evolutionary biologists were quick to point out that this was what they had always believed. Certainly, earlier accounts can be found that accord well with this view. In 1961, for instance, the renowned evolutionist George Gaylord Simpson had written that a species is a lineage, an ancestral descendant sequence of populations evolving separately from others and with its own unitary evolutionary role. Very much the view I've just proposed. But there is a second reason why the species as individuals thesis seemed incredible to some, and that was that a species seemed such a peculiar kind of individual. The parts of a species are entirely disconnected from one another and move independently from one another. Apart from the network of reproductive links, they seem in no way integrated with one another. And of course, all these problems are exacerbated if we allow a species to include a number of isolated populations, like the rabbits in Europe, Australia, and elsewhere. This second problem is quickly solved, however, when we recognize that a species or a lineage is not an individual thing, but an individual process. What we need here is the concept of a continuent process that I outlined at the end of the last lecture, and which is sometimes ruled out by traditional philosophical definitions. There are many examples of processes that consist of spatially discrete, more or less autonomous parts. Think of a battle or a coastal erosion. The separation of a river into distinct streams, on the other hand, parallels the separation of rabbits between continents. Perhaps they will rejoin and continue to be parts of the same river. Perhaps they will take their own separate ways to the sea and be judged to be separate rivers. Having said what I think a species is, I must enter two important qualifications. First, I don't think that all species are processes. As I've already noticed, it is highly questionable whether bacteria or other microbial organisms form proper lineages. I should perhaps clarify, when I said all, that I don't think all species are processes, I mean all kinds of animals named as a species, because there we may be just classifying, not pointing to a process. If they do not, we still need ways to find ways of classifying them, and cladistic methods will not be available. This brings me to my second, more general point. The The word species has, for centuries, been used to name our so-called basal units of classification, or basal taxa. This is the lowest level of classification that is considered biologically significant, and divisions into subspecies, races, and such like are generally considered of minor biological importance. And hence, the answer to the question, what kind of organism is this? Surely, we should not abandon this usage because the word species has come to have a more specific theoretical meaning as well. It is convenient, as is no doubt often the case, if the organisms that form the parts of a lineage coincide exactly with the organisms that fall into the same basal taxonomic unit. But we know from the case of bacteria that this cannot always be the case, as sometimes there is no well-defined lineage to map onto the taxonomic unit. And in fact, given the very different roles that these uses of the word species play, we should not be surprised if there are other occasions where their reference diverge. Ernst Mayr, another leading figure in 20th century evolutionary biology, provided the most widely accepted definition of the species, the so-called biological species concept. According to Mayr, I should say by contrast with other things like the evolutionary species concept or the phylogenetic species concept, all of which obviously are biological, but Mayr managed to capture this rather tendentious description. According to Mayr, a species is a group of organisms connected to one another and isolated from other organisms by sexual relations and thus gene flow. Mayr did not much care for microbes, and since he didn't think they formed species, they didn't provide any problem for his preferred concept of a species. But this is, of course, just to assume the priority of evolutionary theory over the pragmatic demands of taxonomy. In fact, it is best to read Mayr's criterion as identifying species individuals. Sexual reproduction does indeed seem to be the key factor that provides coherence to such individual processes when they exist. But, as I've insisted, there is no reason to assume that every organism is part of a species in this sense, as opposed to being a member of a species in the taxonomic sense. I will make just one further point on this topic. Species, I have said, can be either units of evolution or units of classification. Often the two coincide, that is, the parts of the units of evolution are the same as the instances of the classificatory term. However, classification is a highly pragmatic activity. It is essential for the transmission and storage of all manner of biological information. If I see an animal behaving in an interesting way, I cannot communicate this information usefully without knowing what kind of animal I am observing. To serve this purpose, there is an optimal size of the basal taxon. Mammal would not serve this purpose well, being far too broad, but nor would the division of the capercaillie into 17 distinguishable subspecies. Evolution, on the other hand, has no such prejudices. Lineages may diverge into a large number of fully distinct sublineages, or they may last for very long periods of time, changing dramatically or becoming morphologically highly diverse. Classification and evolutionary theory aim to track different phenomena. On the one hand, clusters of behavioral or morphological similarity. On the other, the divergence or continuity of evolving populations. Insisting on their coincidence may often lead to a very inferior classification. Every organism should, ideally, belong to a classificatory species, but many will not be part of a coherent lineage. Perhaps it would be good to have different words for these very different concepts, but often the members of one and the other and the parts of the other coincide, and, surprisingly or not, the polysemic character of the word species doesn't seem to do much harm. I want now to say a bit more about these species processes and how they evolve. In the previous lecture, I noted that a fundamental distinction between processes and things is that whereas the latter are assumed to be stable by default, for the former, whatever stability they show requires explanation. So, what explains the stability of a biological lineage. A lineage, in the first place, involves the constant production of organisms fairly similar to those extant at the time of production. This is clearly a necessary condition for the persistence of the lineage through time. But to count as an individual process, rather than merely a set of disconnected events of organism production, a species must exhibit some kind of internal organization. For sexual organisms, this may be sufficiently achieved by reproductive links. Reproductive connection and isolation from the introgression of alien genes is often taken as a defining character of a species. For many species, notably our own, there are also social interactions that provide further internal organization. A set of reproductive and perhaps social links between members may seem a rather paltry degree of organization compared to the exquisite organization of an animal. But as David Howell wrote, most organisms do exhibit more internal organism than most species, but this difference is one of degree, not kind. Most species do not exhibit the internal organization common in vertebrate organisms, but the same can be said for plants as organisms. Most plants do not exhibit the internal organization common in vertebrate organisms. So, supposing that we allow that a lineage displays sufficient organization to qualify as an individual process, what sustains and crucially stabilizes this organization? The first and most important answer to this question is natural selection. I should say the second, because I mentioned reproduction, I'm sorry. So, a process perspective highlights the importance of the often underappreciated point that most natural selection is stabilizing. Why do we have a more or less coherent lineage constantly generating organisms of a more or less similar kind? Because the great majority of substantial changes to the organism reduce its fitness, because the organism is an exquisitely organized process, closely aligned with the environment in which it lives, most changes disrupt this organization in some way. Given that most species produce offspring greatly in excess of the numbers required to maintain themselves, there is plenty of opportunity for competition, and the novel will generally lose out. So, natural selection alone may be sufficient to stabilize a coherent species process, at least together with, obviously, reproduction. The importance of stabilizing selection tends to be underappreciated, partly because the idea of natural selection as a creative force often seems more exciting, and perhaps also partly because the philosophical preference for things over processes tends to obscure the great importance of stabilizing processes. Just to emphasize the point, imagine a world in which survival was always purely a matter of chance. Individual lineages would gradually drift away from their starting conditions so that even if somehow we started with a reasonably homogeneous and coherent species, the homogeneity would decrease generation by generation. To use a phrase dating from Georges Cuvier, but also frequently by Darwin, natural selection ensures that the lineage remains within the limits of its conditions of existence. I should also say a bit more about the factor that Ernst Mayr took to be the defining factor for a species, sexual reproduction. To connect with the last point, stabilizing selection is plausibly a necessary condition for sexual reproduction. Without it, variation within the species would inevitably increase, sexual compatibility would decline, and sexual species would eventually become extinct. Perhaps this is the situation in much of the microbial world, where there is constant competition between ephemeral clades and no persistent individual lineages emerge. One way of seeing the importance of sexual reproduction is that it maintains the boundaries between processes. As we saw in my first lecture, processes do not necessarily have sharp boundaries and are often liable to merge or intertwine. But without any limitations to gradual merger with surrounding lineages, it would be impossible to maintain a complex and coordinated suite of adapted traits. Sexual reproduction can function in the way Mayr envisaged to construct and enforce the boundary between lineage processes. Given that the boundary between related processes by no means is a given, it's no surprise that evolution should have provided lineages with various means of enforcing their boundaries. Adequately isolated species survive longer than those that dissipate into their living surroundings. Natural selection, as Richard Dawkins insightfully noticed, and I should perhaps confess that I don't always or even often agree with Richard Dawkins, and I might disagree with him a little later on, but one of his an insight that I think is really important is that its most general natural selection is the survival of the stable. To be stable, a process must have some kind of boundary. The boundaries devised by sexual species are an amazing biological phenomenon. A great variety of isolating mechanisms served to decrease the likelihood of mating with an organism external to the lineage. Isolation may also be achieved ecologically, behaviorally, or mechanically, as in the lock and key morphology of many insect genitalia. They may be temporal via desynchronized life cycles, for example, adult insects or tree pollen appearing at a time distinct from that favored by similar species. If these devices should fail, various forms of genetic incompatibility and hybrid sterility that come into effect after mating provide a second layer of border defense. There is also a positive analog of these isolating systems, so-called mate recognition systems. The exotic color patterns of many reef fish, the elaborate songs of birds, the signaling molecules of many fish and insects, the flashing lights of fireflies, the extraordinary dances of birds of paradise, all likely serve mate recognition purposes. I'm sorry, I missed a click. Sorry. The function of such traits is often difficult to pin down, as such signals are very likely to produce a basis for intraspecific competition for mates, and may even trigger processes of runaway sexual selection. But the importance of mate recognition is not in question. Indeed, it has even been considered so important as to provide the best way to define the species, though I think this is an exaggeration. Boundaries between species, despite all of this, are seldom sharp. Hybridity is common in most domains of life. Depending on the fertility of hybrids, genes may flow freely between species. One classic study on American oaks showed that despite extensive gene flow, species retained strong morphological distinctness, suggesting that natural selection, tracking distinct fitness maxima with a fitness trough separating them, may often be sufficient to counteract the homogenizing effects of gene flow. In other cases, it may be a matter of judgment whether there are distinct species at all. That is often how it is with processes. A final crucial way in which lineages are stabilized is niche construction. Organisms do not merely track the environment, maintaining sufficient adaptation to the current state of affairs. Their activities change the environment and actively construct the environments to which they are adapted. Indeed, very little in biology is one-directional and linear in the way that passive tracking of the environment proposes. Dramatic examples of niche construction are such structures as the beaver dam or the termite mound, but all or almost all organisms make some changes to their environment and hence to the environment to which they are better or worse adapted. A nice case discussed in detail by no less a biologist than Charles Darwin is the earthworm. The earthworm evolved from an aquatic ancestor and never fully adapted to life in a dry environment. However, by cycling organic material into the soil, earthworms are very effective at maintaining the moisture level in the soil to a level at which they can flourish. As all gardeners know, this is also very beneficial to many plants. Niche construction need not be a phenomenon limited to the individual or the family group. The niche may be shaped by countless conspecifics, present or past. In the case of earthworms, it is not even a particular species, but many different species from the phylum Annelida that contribute to the environmental modification that enables terrestrial life for these creatures. This activity continues through time and space and stabilizes multiple lineages. But the most striking example of all is the human. We live in hugely elaborate structures within structures, and our ways of life are wholly dependent on the infrastructure that we create. I shall return to this shortly and later. Sexual reproduction is a common but not universal feature of evolving lineages. Niche construction is, in a sense, more nearly universal, as every organism has some effect on its environment. But the construction of complex structures over many generations of the kind exemplified by termites, beavers, or humans is a quite rare accomplishment. All of these varieties of lineage stabilization, as opposed to merely external pruning by natural selection, can be seen as stabilization through internal organization, a movement along the spectrum of degrees of internal organization described by David Hull. The most extreme move along this dimension is the evolution of sociality. Sociality within a species provides obvious elements of additional internal organization. These have reached their peak in social insects and in humans, in which a complex division of labor is made possible by a system of distinct and interlocking roles. Members of such a species interact with one another in a variety of ways that generally contribute to the maintenance of the complex structure. But social structure also contributes to a rather different feature of a lineage, evolvability. Sociality provides new ways in which lineages can respond adaptively to their environments. To consider these, I must now turn more generally to a more familiar topic in evolutionary theory, the determinants of evolutionary change. Many evolutionary theorists still assume that the sole or major architect of evolutionary innovation and change is natural selection. I'm skeptical of the creative character claimed for natural selection. In one sense, I think it is a simple, logical truth that natural selection cannot be creative. Selection is selection. The choices between which the selection is made must come from somewhere else. The counter-argument, perhaps most prominently defended in recent years by the aforementioned Richard Dawkins, is that provided a very small series of steps can be described leading from the present phenotype to an advantageous future state, small mutations will arise that affect the transition to the next step, and eventually the journey will be completed. Dawkins illustrates this with the case of the vertebrate eye, aiming to rebut the common thought that half an eye is no use to anyone. But why should we believe that a mutation should even be possible to create precisely any envisaged change along such a pathway? And furthermore, bearing in mind that genes typically have many different effects, so-called pleiotropy, and these other effects are very likely to be deleterious, why assume that the envisaged change will be adaptive and therefore selected? We should certainly acknowledge that natural selection, by maintaining stable and integrated lineages, creates the conditions for major evolutionary innovation, but asking it to provide the innovations with no help but small random mutations is very probably asking too much. Enthusiasm for the overriding importance of natural selection has tended to draw attention away from an essential task in understanding evolution. Where do evolutionary changes come from? There are many possible answers to this, and it would be a serious error to assume in advance that any of them is of predominant importance. But let me mention a few interesting options. I should also remind you that these are changes to the developmental system, that is what the organism is, and may occur at any point in the developing process. As well as generally small mutations and recombinations, which are the sources of most interest to orthodox gene-centered evolutionary theory, there are further genetic options. Evolutionary developmental biology, of which more below, has taken great interest in architectural genes in which small changes may produce major morphological effects. It's also possible that horizontal acquisition of genes has had great importance. The mammalian placenta, for example, appears to be constructed from tissues that originated from retroviruses inserted into the genome. Epigenetic changes to the genome, changes that affect the expression of genes without changing the sequence of nucleotides, the components, may be directly inherited or may have behavioral effects that lead to their reconstruction from generation to generation. This remains particularly controversial, in part because it suggests the inheritance of acquired characteristics, which has long been something of a taboo in evolutionary thought. But the evidence for inheritance of epigenetic changes is growing steadily. Changes to parental behavior may be learned by offspring. Where a species has developed some kind of social life, there may be transmission of behavior from non-parental conspecifics. These processes are the basis of what is known as cultural evolution. An interesting case here is that of behavior related to the construction or maintenance of the niche. And this list is by no means exhaustive. The last few suggestions are not available to all species, a point the importance of which I shall elaborate shortly. I might also mention, if only in passing, a fascinating topic in evolutionary theory, the possibility that complexity can arise from undirected drift, so-called constructive neutral evolution. The extension of these sources of variation is sometimes thought of in terms of the extension of modes of inheritance beyond the familiar genetic. Inheritance methods might be thought of as providing vertical or temporal stabilization of the lineage as opposed to the stability at a time provided by, for example, social or niche structure. But they also provide spaces for evolutionary change. While epigenetic inheritance significantly extends the possibilities allowed by orthodox neo-Darwinism, sociality opens up entirely new inheritance pathways. A minimal but very important form of sociality I mentioned is parental care. This surely qualifies as a kind of sociality in the sense of structured relations between species members with, in this case, distinct roles. A general term for a range of inheritance processes made possible by parental care is parental effects. Parents constantly interact with their developing offspring, and these interactions can have important effects on offspring development. This is too familiar from human experience to need much argument, but it is by no means unique to humans. Cultural inheritance in social species can extend far beyond the parental. In fact, one of its most distinctive features is that transmission of culture can happen between arbitrarily selected species members. Again, this is most obvious in humans, but a wealth of observations on such issues as tool use or foraging choices shows that it is a common feature of many other species. There's a long tradition of theoretical work on the dynamics of cultural evolution, but curiously, it remains common for evolutionary theorists to insist that it is a phenomenon of only marginal importance and has little relevance to the theory generally. This is a good point to contrast what I've been saying about evolving lineages with a common but simplistic understanding of evolution that remains influential. This is the idea deriving from the so-called modern synthesis of the mid-20th century that evolution is entirely, or almost entirely, a matter of changing frequencies of genes, which have typically small effects on the organism, and which spread through populations by natural selection. I've mentioned this picture in discussing the idea of natural selection as creative, and specifically in relation to the writings of Richard Dawkins. In Dawkins' work, this idea is associated with the suggestion that genes are selfish, which, interpreted charitably, means that genes are the drivers of evolution, the things that are selected on the basis of their effects on the essentially passive organism in which they reside. Much of the dismantling of this rather simplistic story has come about under the rubric of evolutionary developmental biology, or evo-devo. Evo-Devo recognizes that it is the life cycle that evolves, and then is well positioned to observe the significance for evolutionary theory of the fact that the determinants of development are complex. This last topic is more strictly associated with the school of developmental systems theory. Both these movements in evolutionary biology depart from the gene-centered orthodoxy by recognizing the fundamental role of the individual organism in evolution. Moreover, they recognize that the organism itself must be seen as a developmental system. Both, in slightly different ways, have looked at the developing system as the fundamental unit that evolution has produced. They are, therefore, if not always explicitly, process theories. While I'm generally entirely sympathetic to these perspectives, in this lecture I focused not on the individual developmental system, but on the larger and longer-lasting entities composed by these developmental systems, lineages or species. In the final part of this lecture, I shall make some rather different remarks about these remarkable phenomena. Let me return to the two senses of species. In the classificatory sense, every organism belongs to a species. If we find an organism that we cannot assign to an existing species name, we invent a new one. Every organism, in this sense, belongs to exactly one species. But the species as a material entity, a continuent process, is something quite different. I said earlier in this lecture that the lineage or species was what evolved, but it is not merely, as I stated then, that the distribution of properties among its members changes as it evolves. The nature of the process also changes, and the highly integrated species that occur in social animals are themselves the highly integrated species that occurs in social animals is itself a highly evolved entity. Not all species in the taxonomic sense are integrated or stable lineages. As I mentioned earlier, it is questionable whether bacteria, or I should mention archaea, the other great group of nucleus-lacking microbes, belong to integrated lineages at all. And henceforward, when I say bacteria, I will take this to include archaea. The lineage of a bacterium may simply be the sequence of its ancestral cells, and classification of bacteria may be based on quite different factors. It's likely that natural selection will maintain loose populations of similar cells, but these may be of brief duration. Bacterial taxonomy was originally grounded in pathological properties, though this covers a minute proportion of the real diversity. More recently, various measures of genetic similarity have been used. It's also been suggested that most bacteria do exist in something surprisingly similar to the sex-based species by virtue of exchange of genetic material within a restricted group. But no one claims that this is true of all bacteria, and even if it were, there is no reason to assume that these groups would provide a universally optimal ground for classification. More to present purposes, the sociality, parental relations, and so on that I've been discussing do not apply to such species, though in the next lecture I discuss respects in which bacteria might seem highly social. At any rate, it is controversial whether stable species exist at all in bacteria, and evolution may occur only in very transient lineages. In the case of multicellular organisms, we can note an important distinction, if ultimately a matter of degree, rather than kind, between what have sometimes been known as R-selected and K-selected species. R-selection is the production of sometimes enormous numbers of offspring and leaving them entirely to fend for themselves. The prize winner in this respect is said to be the common molar or ocean sunfish, which lays about 300 million eggs. And I note this contradicts my slide, which has the oyster at 500 million a year. I think that's an exaggeration, but it's not terribly important. The more familiar cod can produce about 5 million. Many insects also produce very large numbers of eggs. Obviously, the overwhelming majority of these fail to reach maturity, but they do provide the opportunity for a lot of natural selection. In such species, there is little integration of the species beyond natural selection and mechanisms for maintaining reproductive isolation. Among vertebrates even, amphibians such as frogs may lay thousands of eggs, and reptiles may lay dozens. Again, there is usually little parental attention. K-selection is the move to high investment in a small number of offspring. This reaches its peak with birds and many mammals, invariably exhibiting a good deal of parental care as mandated by breastfeeding or nest feeding, and often a wider social structure. The lineage in these cases is not only more strongly integrated, but provides new evolutionary possibilities by facilitating a range of parental effects and cultural transmission. Local concentration and cooperation also make possible the most impressive kinds of niche construction, such as exhibited by beavers and humans, or, in a rather different evolutionary trajectory, the social insects. Having mentioned cooperation, I should make a very important general point here. Even evolutionists often like to talk about competition, and no doubt this has been a vital driver of evolution. But as evolution progresses beyond merely the changing properties of the individuals that compose a lineage to changing properties of the lineage itself, cooperation becomes an increasingly important element of the evolutionary process. Cooperation enables the transmission of information between unrelated individuals, thus fostering far more effective cultural evolution. It may allow more effective care of offspring through recruitment of helpers, and it allows far more elaborate transgenerational ventures in niche construction. No doubt, as traditional advocates of the overwhelming predominance of competition will object, there will be some conflict between selfish individuals and the interests of a cooperative society. But the presence of highly cooperative species shows that this conflict can be overcome. No doubt one reason for this is that cooperation can be effective in identifying, isolating, and sanctioning individuals who attempt to advance their own interests over that of the social whole, or at least used to be the case. The success of cooperative lineages is sufficiently demonstrated by the extraordinary successes of social insects and humans. Humans and their livestock make up over 95% of the vertebrate animals on the planet. And for every human on the planet, there are said to be 2.5 million ants, much smaller than us, but a lot of ants. These groups point to a final and perhaps decisive benefit of cooperation, the enabling of a division of labor. This leads me to say a few words on the human lineage, which will be my primary focus for the remainder of these lectures. And here I shall shamelessly espouse human exceptionalism, not perhaps as a matter of kind, but certainly as a very large difference in degree. The human lineage is unique in the degree of its integration and in the complexity of the intrastructural niche it has constructed. A huge part of what has made this possible is the extreme and flexible division of labor that is unique to humans. And a topic to which I will return in the fifth of these lectures, what makes this division of labor possible is the unique developmental plasticity of humans with respect to their behavioral capacities. Just reflect for a moment on the variety of peoples whose efforts have jointly made possible this event today. Some that come to mind are architects, builders, sound and lighting engineers, IT specialists, clothes makers, the designers, manufacturers, and operators of the aircraft on which I arrived in Edinburgh, likewise for the taxi in which I came from the airport, the hotel in which I'm accommodated, and on and on. These roles constantly change, moreover, as typewriter makers and switchboard operators cease to exist and are replaced by bloggers and computer programmers. Of greatest relevance to our present concern, this has created a situation in which genetic bases for evolution have become far less important than social and culturally mediated processes of change. And I don't mean to imply that the former may not also remain important. The schools, television programs, books, universities, and so on that shape the development of humans today are very different from those that did so a few decades ago. Now, for better or worse, we even have professional influencers. And the technologies that have emerged over the last 2 centuries have changed profoundly who we are. The mobile phone that has existed only for a quarter of a century is now as standard a piece of human equipment as shoes or wallets, and far more so than watches. I would argue that by making friends and family instantly accessible at any time and in any place, it has wholly transformed our experience of social space, which no longer coincides with geographical space. The dominant position of humans on our planet, I suggest, is as much the result of the evolution of the lineage itself as of the individual organisms of which it is composed. And it's ironic in the light of this that an ideal of autonomous individuality has been so powerful in recent centuries. For there is surely no doubt that it is the evolved capacities of our species as such, not of individual organisms, that is our peculiar strength. I shall return to related thoughts at several points in the coming lectures. But for now, let me conclude. Over these first two lectures, I've discussed two of the most important kinds of biological system, the organism and the lineage. Without denying the enormous interest of lower levels of organization, such as the cellular, molecular, or genomic, I believe that these enable rather than determine the activities of the more complex systems I have discussed, and so they do not require detailed attention for my present concerns. Organisms and lineages, I have argued, are persistent or continuent processes, an ontological category that has been insufficiently stressed by philosophers. They are also highly interdependent processes. Lineages are composed of organisms and persist because organisms reproduce, but organisms are generated by lineages and have the properties they do because of the particular evolution of the lineages to which they belong. A feature of processes that I have occasionally emphasized is their lack of sharp boundaries and the consequent possibility of their being deeply and even inextricably intertwined. In the next lecture, I shall pursue this theme with regard to the entanglement of organisms, first in connection with the ubiquity of symbiosis in the living world, and second with respect to the gradual disentanglement and divergence of processes in reproduction. But for now, thank you for your attention.